Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Cefsulodin Irgasan Novobiocin, CIN Agar. Table of Contents. Introduction to CIN Agar. Objective of CIN Agar. Principle of CIN Agar. Composition of CIN Agar. Preparation of CIN Agar medium, high media. Quality control and colony characters on CIN Agar. Precautions. Limitations of CIN Agar. Introduction to CIN Agar. Cefsulodin Irgasan Novobiocin. Agar, CIN Agar, commonly known as Yersinia selective agar, is a highly effective selective and differential culture medium designed for the isolation and identification of Yersinia species, particularly Yersinia enterocolitica. Originally developed by CIN D.A. in 1979, CIN Agar features a blend of selective agents, including Cefsulodin, Irgasan, Novobiocin, crystal violet, and bile salts, alongside essential nutrient components. Over time, modifications to its formulation have improved its efficiency for microbiological applications. Objective of CIN Agar. The primary objective of CIN Agar is to facilitate the selective isolation and accurate identification of Yersinia enterocolitica from clinical fecal samples and various food specimens. Principle of CIN Agar. Cefsulodin Irgasan Novobiocin, CIN. Agar is a specialized medium containing selective agents and antibiotics that inhibit the growth of most gram-positive and many gram-negative bacteria, while enabling the identification of Yersinia enterocolitica colonies. Crystal violet and sodium deoxycholate, also known as bile salt, present in CIN Agar, effectively suppress the growth of gram-positive bacteria. Irgasan further enhances this inhibition, also targeting fungal growth. The antibiotic combination of Cefsulodin, Irgasan, and Novobiocin selectively restricts the growth of a wide range of gram-negative and gram-positive bacteria, but allows Yersinia enterocolitica to flourish, helping it outgrow competitors. Mannitol, a fermentable sugar in the medium, is utilized by Yersinia species to produce organic acids, lowering the pH of the medium. This pH change is detected by neutral red, a pH indicator incorporated in the agar. As a result, Yersinia enterocolitica ferments mannitol and produces characteristic bullseye colonies, central dark red-pink zones surrounded by a translucent outer ring, aiding in the identification of this pathogen. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Composition of CIN Agar. The original composition of Siemens CIN Agar, as described by CIN D.A. in 1979, CIN D.A. 1979, synthesis of a selective agar medium for Yersinia enterocolitica. CAN Journal of Microbiology. 25-1298-1304 is as follows. Components and quantities. Special peptone, oxoid, 20 grams. Yeast extract, Difco, 2 grams. Mannitol, 20 grams. Pyruvic acid, sodium salt, 2 grams. Sodium chloride, 1 gram. Magnesium sulfate, 7H2O, 10 milligrams, 10 milliliters of 0.1% stock solution. Agar, oxoid number, 4, 12 grams. Irgasan DP 300-10 milliliters of 0.04% in 95% ethanol. Bile salts, Difco, 2 grams in 200 milliliters distilled water. NaOH, 1 milliliter of 5N-NaOH solution. Neutral red, 10 milliliters of 3 milligrams per milliliter solution. Crystal violet, 10 milliliters of 0.1 milligrams per milliliter solution. Cefsulodin, 10 milliliters of 1.5 milligrams per milliliter solution. Novobiocin, 10 milliliters of 1.5 milligrams per milliliter solution. Final pH, adjusted by 5N sodium hydroxide. 7.4. Reference: Shimon, D. A. 1979. Synthesis of a selective agar medium for Yersinia enterocolitica. CAN Journal of Microbiology. 25. 1298. DOI 10.139/M79-205. PMID 540,256. Modern composition, high media. In modern formulations, companies supply CIN Agar as a base medium, Yersinia Agar base, with separate selective supplements, Cefsulodin Irgasan and Novobiocin, as these antimicrobial components cannot be autoclaved like other ingredients. High Media Laboratories also provides CIN Agar this way, offering the Yersinia Agar base and CTN selective supplement. When mixed in the correct proportions, these components form CIN Agar for selective isolation of Yersinia enterocolitica. Components of CIN Agar per 1,000 milliliters: peptone, 20.00 grams; yeast extract, 2,000 grams; mannitol, 20.00 grams; sodium pyruvate, 2,000 grams; sodium chloride, NaCl, 1,000 grams; magnesium sulfate, 0.01 grams; sodium deoxycholate, 0.50 grams; neutral red, 0.030 grams; crystal violet, 0.001 grams; CTN selective supplement, FD034, 2 vials; agar, 12.50 grams; final pH, 7.4, plus or minus 0.2 at 25 degrees Celsius. Reference: High Media Laboratories. Components per 1 vial of CTN selective supplement, FD034. Cefsulodin, 7,500 milligrams; Irgasan, triclosan, 2,000 milligrama; Novobiocin, 1,250 milligrams. Reference: High Media Laboratories. Preparation of CIN Agar medium, high media. Suspend 58.04 grams of Yersinia Agar base, or the required amount of each listed component, in 1,000 milliliters of distilled water in a suitable conical flask or glass bottle. Mix thoroughly using a magnetic stirrer or by manual shaking. Heat the mixture to boiling to ensure that all components, including the agar, dissolve completely. Sterilize the medium by autoclaving for 15 minutes at 121 degrees Celsius and 15 pounds pressure. Allow the medium to cool to approximately 45 degrees Celsius. For the selective supplement, add 2 milliliters of sterile distilled water and 1 milliliter of ethanol to each vial of CTN selective supplement, FD034, and gently shake to mix. Prepare 2 vials in this manner. Once the basal medium has cooled to around 45 degrees Celsius, add the contents of the 2 prepared CTN selective supplement vials to the medium. Mix gently to ensure uniform distribution, forming the final CIN Agar medium. Dispense the required amount of the CIN Agar medium into Petri plates or test tubes and allow it to solidify completely before use. Shelf life and storage conditions. The prepared CIN Agar medium can be stored at 2 to 8 degrees Celsius in a sealed container for 3 to 8 weeks. Unprepared media components should be stored at 10 to 30 degrees Celsius in a sealed container until the expiration date indicated on the label. Quality control and colony characters on CIN Agar. The unprepared CIN Agar medium powder with neutral red indicator will appear yellow to pink in color, while the prepared and solidified medium in a Petri dish will have an orange-red or pink hue. The pH of the medium should be 7.4 plus or minus 0.2 at 25 degrees Celsius. cultural characteristics of bacteria after 24 to 48 hours of incubation at Joe 22 to 32 degrees Celsius. Escherichia coli, ATCC 25922, shows no growth or partial inhibition. Pseudomonas aeruginosa, ATCC 27853, also exhibits no growth or partial inhibition. Proteus mirabilis, ATCC 25933, displays no growth or partial inhibition as well. Salmonella typhimurium, ATCC 14028, similar results, no growth or partial inhibition. Enterococcus faecalis, ATCC 29212, shows no growth. Staphylococcus aureus, ATCC 25923, also shows no growth. Precautions. Do not add the CTN Selective supplement before autoclaving or when the medium temperature is above 50 degrees Celsius or below 40 degrees Celsius. Use sterile distilled water to hydrate the CTN Selective supplement vial. Ensure the correct amount of CTN Selective supplement is used to prevent the growth of unwanted bacteria. Limitations of CIN Agar. Other Yersinia species, such as Y. Pseudotuberculosis, Y. fredericksenii, Y. christensenii, Y. intermedia, and others, may also grow and produce similar colonies. Bacteria like Aeromonas, Serratia, liquefaciens, Citrobacter frundii, and Enterobacter agglomerans can grow and exhibit similar colony characteristics. CIN Agar provides only a presumptive identification of Yersinia Enterocolitica. Confirmation should be performed using biochemical or genetic analysis. Applications. CIN Agar is used for the isolation and identification of Yersinia Enterocolitica strains from clinical, fecal, samples, and food samples. Safety and disposal. Dispose of used medium by autoclaving or incineration. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you.